If you live off of another organism and you harm that organism in the process, you are a parasite. Up here on land, we can be infected with a gruesome host of parasites, from microscopic protozoans to lice and fleas, all the way up to 3-meter-long tapeworms. Down in the deep, life is so weird already, and parasites take it to a whole new depth. I'm here at the Mullineaux-Benthic Ecology Lab at Woods Hole Oceanographic Institution to meet some experts in deep-sea parasites.
So my research focuses on parasites at deep-sea hydrothermal vents, which are these hot springs on the seafloor where life is just flourishing around them. And it turns out we find a lot of the same types of parasites in the deep sea that we find in a lot of other marine ecosystems. So obviously the pressure and the depth and the lack of sunlight is no barrier to parasites persisting and thriving in these ecosystems.
Parasites are not all bad. In fact, they're pretty incredible ecosystem engineers, helping to keep populations from getting out of control, which can allow other rarer species to survive.
There's this crazy parasite called a rhizocephalin, which is actually a type of barnacle, but it actually attacks and invades crabs and other crustaceans, and it castrates them, and it grows a rootlet system through the host's body. It still looks like a crab, but it's basically just producing parasite offspring. So it is sort of the parasite's own personal factory. These truly are the things of horror movies. Sometimes the parasite can change the host's behavior in order to make it more likely to reach its next host. These are just wild ways to live. And the fact that this evolved and then is persisting and working, especially in an environment as harsh as the deep sea, I think is really interesting. And it helps us get at what are the limits to life persisting and surviving under harsh conditions.
Yeah, absolutely.
Roundworms, or nematodes, were found almost 2 miles deep in a gold mine in South Africa, somehow surviving on bacterial biofilms near to drilling holes. Finding life this deep opens up the door to what life might exist on other planets, especially those which might previously have had conditions for life but now seem uninhabitable.
So one of my favorite parasite types is called the trematode. These are those parasitic flatworms. And what interests me about them is they don't only use one host species, they have to pass through 3 different host species to complete their life cycle. I thought in ecosystems like vent in particular, where the habitat is really patchy and it's also really disturbed by volcanic eruptions, I thought a parasite that needs 3 hosts to be there, especially in a disturbed system, might find it hard to live there. And that's actually not what we found. There are a lot of trematode species at vents, and there seems like they're completing their life cycle entirely in vent hosts. So trematodes start as an adult in the gut of a fish, and then their eggs are passed out of the fish and hatch into this swimming larva called a miracidium. And this is microscopic. It swims through the water and it finds the next host, which is a snail. And inside that host, it makes these asexually cloning sacs. And it actually castrates the snail, just like that rhizocephalin that we were talking about earlier. Then these asexually cloned larvae basically leave the snail as these free-swimming forms called cercaria, and they penetrate the next host and form this cyst called a metacercaria inside any number of species. It can be a small crustacean or a worm or even a small fish. And that cyst just lies in wait in that host, and it's waiting for that host to get eaten by the predator, which is this big fish. These parasites rely on so many other species to persist. They can be indicators of an ecosystem that is diverse and has healthy functioning interactions between species.
I guess, yeah, it's probably a really good thing that this view around parasites is starting to change a bit, because as wacky and weird as they are, as uncomfortable as they make us, they are obviously very important for the ecosystems that they live in. So in order for the ecosystem to thrive and be healthy, I guess parasites have to thrive and be healthy as well. So I think it's probably fair to say that deep sea parasite research is a pretty niche field, but I know that over in Corpus Christi, Texas, a friend of yours, Dr. Chuck Blend, he's waiting on the line for us, right?
Yeah, my friend Chuck is actually about to do some live investigations for parasites right now, so why don't we go watch?
Absolutely, let's do it. So I see you've got some super interesting equipment here, and I'm very, very excited about it. So what is it exactly that you're going to be showing us today?
Well, today we get the opportunity, everyone, to join me in a real live deep sea fish examination for parasites.
Take it away, let's do it.
All right. Well, I have brought here a container of deep sea fish. Specifically, these are called tripod fish. They're on the abyssal plains and continental slopes of oceans throughout the world, and they have modified fins to be able to stand up off the bottom just a few centimeters. And because these fish are so hard to obtain, the chances are very good that what we find today will be new to science. I've not examined tripod fish before, so this is first time for me too. I'm really excited to see if this particular individual has any parasites in or on him. I'm going to lift up the gill flaps and check the gills. So this fish is too healthy. Nothing so far. All right, so the external examination is complete, but the good stuff is yet to come as we go inside now. So I'm going to make an incision, get just above the stomach, because the worms I'm looking for are in the GI tract there, gastrointestinal tract. Now I'm going to go through the internal organs, and I'm going to separate them out. The gonads, the liver, and I'll set that aside. I want to go through now and look at the intestinal tract.
We always go through each organ individually because some parasites specialize on different organs, so it gives us really good data on what habitat the parasite needs inside the host.
Ooh. Okay, now there's something that pops out. I don't know if you can see it. This looks like possibly a roundworm. And I will set this nematode aside and look at it closer after the examination. Oh, well, what do we have here? This looks promising.
Could be new to science.
I have found acanthocephalans, those spiny-headed worms I was telling you about. There are several in here. They have a head on them called a proboscis. Imagine a baseball bat with lots of nasty-looking spikes and hooks on them. Basically implant themselves, anchor themselves in the wall, the intestinal wall of a host. And acanthocephalans use arthropods as intermediate hosts. On land, that's insects, and in water and aquatic environments, that's crustaceans. I hope you can see this. There they are.
I see them.
Yes.
And that's the acanthocephalans, and you can see one of them, at least one, has little proboscis sticking out, that spike-covered baseball bat. I was looking for these. This is wonderful, wonderful. I can see it right there. This is a type of fluke, and I've nicknamed it gunslinger, because if you look at it, it has two organs called vitellaria. They produce yolk for the egg, and it looks like a gunslinger. This is going to be something new, folks. Now, when you find one, there's typically going to be some more. We got another one. I have what looks to be at least one other fluke. The fact that we have more than one individual worm allows us to measure that variability, which is important. It tells us a lot of information. I'm going to remove it and put it with his friend in here. And sure enough, that's a third fluke. Now we have 3 from this fish. All right, one last look, and then I will finish up just scanning the ovaries and other connective tissue, liver, things like this. Oh, it's pretty good haul. 3 digenetic trematodes, 3 acanthocephalans, and a nematode.
Once at sea, I was dissecting event fish, which are much bigger than the fish that Chuck is dissecting, so it's a lot to process.
Oh, yeah.
And I found 500 trematodes in its stomach, and I pulled out every one individually. I was up all night.
I was gonna say, I can't even imagine how long that would take.
That completes the examination of this wonderful fish who produced for us 6, 7 parasitic worms, and chances are 9 times out of 10, this is something new to science. And so, congratulations, everybody.
Yeah, woo, amazing. Even though I am still a little bit grossed out by parasites, I have definitely a newfound respect for them, and this is so amazing. So thank you both so much. So there you have it. This is kind of the perfect example of how little we actually know about our oceans. The more we look, the more likely we are to find something that is completely new to science.
You probably already know this, but whales are incredibly noisy creatures, using tons of different clicks and squeaks for purposes that we believe are probably navigation and/or hunting. Much like myself, whales are also highly social communicators, so all these wacky noises are a vital part of keeping the pod together. And the males of just a few species, including humpbacks, even sing. But new research shows that these males actually learn these songs from each other and can pass them halfway across the globe. Ellen Garland is an expert on this, and she's joining me from Scotland. I remember learning as a kid that whales could sing, and I always thought that it was super cool, but reading even more about your research specifically is absolutely fascinating. It kind of puts whale song in a cultural context, right? So can you tell me a bit about that?
So we're trying to understand how and why humpbacks are actually singing, and then how they're learning these different songs and how they're passing them across this really vast region.
So just for clarity, can you explain what you mean by passing it through different regions?
So each population sings their own song each year, but the song is constantly evolving, it's constantly changing, and all the males in the population will accumulate, they'll learn those changes, and the song moves forward in small to large steps. But what we found in the South Pacific is those songs, those whole song types, are then passed to the next neighbouring population, and they learn it really rapidly and really quickly, and then it's passed to the next one and the next one. So we have this whole large chain of populations where songs are passed from populations in the west to populations in the east, and it's their entire song display. So they'll be singing this song, it's great, and then a brand new one will come in with a completely different arrangement, and they'll abandon that current display for this brand new display really rapidly, and it's an insane cultural change.
I almost want to compare this with our own human fashion trends, you know? In the 90s, everyone's wearing one thing, the 2000s come along, and we adopt a whole new style, and like with whales, it's kind of our way of finding a mate too, right? Having good sense of style?
It's a great analogy. A really cool new outfit comes in and everyone wants to wear it and wants to be seen in it, and then another one comes in, and it's just these waves of these cultural changes. The humpback song has all the hallmarks of a sexually selected signal, so whether they're displaying to a mate, whether it's to do with male-male competition, or both in a multi-message signal framework. So if you're singing something new, these new songs just jump out, they really stand out. So you would assume that a female, they're going to definitely hear these differences, and what that conveys to a potential mate, well, you know, that's a massive question.
Now, I understand that the term culture can mean a couple different things, so can you explain what you mean when you say culture in this context?
I define culture in a really broad sense as the shared behaviour or information within a community which is acquired through some form of social learning from your conspecifics.
So what we're talking about here is whales learning new songs, potentially from parents or peers.
Absolutely. So parents to offspring is actually vertical cultural transmission, and peers is horizontal transmission. We don't know exactly, of course, the function of the songs and what content is in there, and obviously this is a population identifier, so there's information on that. It's information that it's a mature male, and obviously he would be ready to mate. So there's that sort of is the information thus far I think we can safely talk about as part of the song.
Now, I understand you have some new research coming out. That is very exciting, so can you tell us a bit about that?
During my PhD, we traced the cultural transmissions of these song types across the South Pacific. So songs started on the east coast of Australia and were passed in a sequential pattern across to French Polynesia, which is in the central South Pacific. And we looked over a decade, and there was this unidirectional pattern. It always went east. And we know from previous research and other researchers that songs are passed from the west coast of Australia to the east coast of Australia. So we have this huge transmission network from the west coast of Australia across to the central South Pacific. And the next piece of that puzzle is to look from the central South Pacific to the west coast of South America. And so we have traced the song types spreading from French Polynesia in to the west coast of South America into the Ecuadorian population. So now we have song types that will spread across the entire South Pacific and almost around half the globe. It's in a phenomenal cultural connectivity network we've got going here.
To hear that humpback songs are learned and then passed right across the Pacific is just fascinating. What's the next thing you're hoping to find out?
To learn a new song type, you've got to be in acoustic contact. You've got to be able to hear each other. And we think with humpback song, it only transmits tens of kilometers in the water. So it's not going the full ocean basin. So there's obviously some connections, perhaps on the Antarctic Peninsula, but trying to understand how these populations across this vast region are actually interacting. Just because they're sharing song doesn't mean they're potentially breeding, or are they? So it's a lot of interesting sort of population understandings going on there. But also, does the song continue? Do we have this huge full circumpolar transmission around that southern hemisphere?
So what I'm also wondering is, can this research be used to help us protect whales?
In the South Pacific, we still have what are classified as endangered populations. So understanding the connectivity between these endangered populations is really important. Song seems to be a really sensitive indicator of these connection events, of these movements and potential meetings between the whales. So it's may help feed into some of these conservation measures.
We just touched on how Ellen's research can help protect whales, and now I'm off to meet Sophie Van Paris, whose team is trying to do exactly that, by listening for whales just along the coast.
Hi, Sophie.
Hi.
It is so wonderful to meet you.
Wonderful to meet you too. Come on in.
Thank you so much.
So I'm Sophie Van Paris. I run the Passive Acoustics Research Program here at the Northeast Fisheries Science Center. Right now you're in one of our acoustics labs with all the equipment that we use to do our research.
I've been hearing a bit about how complex whale cultures can be and how vocal they can be as well, but I understand that you're listening to whales for a totally different reason, right?
Ship strikes are lethal to whales, can often be lethal to whales, and they're one of the two main sources of whale deaths. The other one is entanglement and fishing gear. And so ship strike is a real major concern, especially for the North Atlantic right whale. That's a critically endangered species.
Absolutely.
Yeah.
So you have a whole network of hydrophones along the Atlantic coast of the US listening for whales and ships and all of these other noises. So do you have one that we can take a look at?
Absolutely, for sure. So there's two different types of recordings that we make.
Right.
We call them archival. When we throw something like this out into the bottom of the ocean, we anchor it down with a weight, and when we want to go back and release them, we send a signal to them underwater. And so this will pop up, and our acoustic recorder, which has been down in the ocean for about 5 months, and this is the hydrophone here, recording everything it hears, will come to the surface.
So what exactly are the kinds of things you're picking up using these hydrophones?
Everything. So we listen to what we call soundscapes. Any whales that are making sounds, any seals that are making sounds, any fish that are making sounds, shrimp even make sounds. You can hear boats, you can hear underwater explosions or seismic activity, and you can hear rain, hear underwater earthquakes. So we listen to everything and anything that happens underwater. So we'll have some in the Gulf of Maine listening for right whales because they're endangered and there's a big problem there. We'll have them in wind energy areas prior to when there's going to be turbines that are being built. But in terms of ship strike, we tend to use recorders that are at the surface and not down at the bottom. So they're not archival, they're called real-time. So those real-time ones we tend to also put in areas where we know there's a lot of shipping. So it'll be in a shipping lane or in an area where there's going to be concentrated vessels active. So what happens is once you hear a sound, there's a little computer on board that actually is going through listening for different sounds of different whales, and once it hears them, it'll send that information up via satellite. It then goes to one of our research assistants in the group. She'll check it just to make sure the computer didn't make a mistake. And then what we do is we send that information off to our regional office. And they will push that information out and put in place things that are called these dynamic management areas. They're really kind of these boxes that you draw around the sound that are put in place, and they warn mariners that whales are there, particularly North Atlantic right whales, and they need to slow down.
So if we get the ships to slow down, how much of a difference does that really make?
There's been a bunch of studies that have showed that slowing down, particularly to under 10 knots, whether it's a large ship or a small ship, can make a real difference in terms of the ability of the whale to survive the strike.
Okay. And so I would imagine that with very few, I think around 350 North Atlantic right whales left on Earth, that this is probably something that's really important to help protect them, right?
Absolutely. Anything we can do is going to be important to help protect them, particularly with ship strike. If the boat's going too fast, it can be lethal. It can really injure the animal, and we really don't want that to happen. We have too few of these North Atlantic right whales out there.
Sophie, thank you so much for speaking with me today. Your work is absolutely inspiring.
There you have it. Whales learn songs just like we do, with people like Ellen tracking how the songs are changing and evolving. And that, in turn, helps people like Sophie find ways to better protect them and potentially save them from extinction. If you watch lions or orca coordinating a hunt, or a family of chimpanzees socializing, we as humans can relate to what they're doing. Their calls and their body language aren't all that dissimilar to what we as humans use. But squid and octopus are cephalopods, and estimates are we haven't shared a common ancestor with them for over 500 million years. Yes, that's billion with a B. Fun fact, the word cephalopod literally means head foot. Cephalopod. Which makes a bit of sense, because octopus and squid are basically all head and feet. Well, arms, not feet, but you get the idea. Anyway, many octopus and squid have this incredible ability to change color for camouflage. But they also change color to communicate with each other and other species. Researchers have been trying to figure out what they're saying and how they're saying it since the 1800s. Now let's take all the difficulties of trying to understand cephalopod communication, and let's pretend that your study species is fast and aggressive and lives deep in the middle of the open ocean. I've got Ben Burford on the line, who's been researching just that. So I'm really interested in your research about Humboldt squid, but just to remind everybody, these aren't just little squids that are splashing around in the shallows, right?
These are really incredible animals that I have a lot of respect for. Yeah, the Humboldt squid is an animal that really never comes into contact with a hard surface in its life. It's what we call an open ocean animal. It lives, breeds, and dies way out at sea.
Humboldt squid have large eyes and excellent sight, and like all squid, they use jet propulsion to swim. This makes them fast, efficient, and highly effective hunters. But Humboldts live fast and die young, growing to 5 feet or longer, but only living for around a year. I find it absolutely fascinating that they live in an environment that most humans will never come into contact with. I mean, as much as I'd love to come into contact with the deep sea, it's just not really possible for my own physical body.
Yeah, it's also the largest inhabitable space on our planet. I mean, the deep ocean and the open ocean comprise, over 93% of our biosphere, the inhabitable space on our planet. And these are animals that specialize in living in that environment. And trust me, it's an environment that you and I would quickly perish in.
So I understand, like other squid, they use colors to communicate with each other. But as we've said, they spend a lot of time in the deep sea where it's basically pitch black. How does that work?
The first paper that really described the variety of color patterns that we see in species of deep sea squids came out, and this was a shock. It's not like a coral reef where there's all sorts of structure and color and things like that. No, it's a dimly lit, empty habitat for the most part. That work really motivated me to think about, well, what do these squid use? They're a little bit different than the classic cephalopod paradigm for communicating with pigmentation patterns. Your classic shallow water cephalopod or squid is essentially something like words on the page of a book. You need light in order to read them. But Humboldt squid and some other deep living squids we think are slightly different. We think they're more like e-readers. You don't need light to read an e-reader because the words on the e-reader are backlit.
I guess I was kind of having a hard time fully understanding how exactly you see something in the dark, but kind of backlighting it makes perfect sense.
The way that Humboldt squid backlight these patterns has to do with their muscle tissue, which is right beneath their squid. It's just like you or me. And for the Humboldt squid, embedded within their muscle tissue are hundreds and hundreds of these tiny little bioluminescent organs called photophores, like a firefly. That is a photophore that's designed to project light out, like a flashlight. Humboldt squid, on the other hand, they're more designed just to radiate light in the tissue itself. And so you can think of these animals as having like hundreds and hundreds of teeny little Christmas lights sprinkled throughout their muscle tissue that like cause their body to glow. They have regions that we think would be brighter than others based on the density of these photophores, and then it selectively conceals and reveals different parts of a glowing body.
We have this image here that's showing 28 different patterns that you've observed of these animals communicating with each other in the wild. Can you tell us kind of what's going on with those different patterns?
That's what we set out to figure out. Were any of these patterns associated with certain contexts? A squid trying to catch food, or a squid in a super dense group. I mean, these animals live in groups. You rarely find them alone, and so presumably some of these patterns may serve to coordinate their group functions. Living in groups is a huge adaptation for life in the open ocean, where resources are very patchy. These squids may have to search for a long time before they find, say, a food resource. But for a group to work, when you find that food resource, perhaps you need some way of conveying your intent, or like what you're doing in the moment that you're feeding. Humboldt squid are highly cannibalistic. Research suggests that they're the most cannibalistic of all cephalopods. They're really aggressive. We actually observed many feeding scenarios where there would be hundreds of squid all just zooming all over the place, catching fish, catching shrimp. And you could think of these patterns kind of like turn signals and brake lights on your car. When you're driving in heavy traffic, which is super, super dangerous, you need to convey to others around you what you're doing. If you want to change lanes, put on your turn signal first and then change lanes, right? So same idea with the squid. If you're around a whole bunch of 2-meter long squids that are all very, very hungry and you spot a fish and you're gonna go for it, you might want to convey to the rest of them, like, I'm gonna go for this fish. But there's also other things these patterns can be used for too. For instance, camouflage. Simple counter shading is one of the patterns we observe all the time, where they're darker on the surface of their body that faces the sun or the surface.
I guess my next question is, how close do you think we are to understanding exactly what they're quote unquote talking about in these groups?
The key thing that we need to do is to establish cause and effect. If presented with a specific color pattern on another squid, how does a squid respond? With Humboldt squid, that'll be a little bit trickier because they live in this really hard to access environment down in the deep sea. Being able to project a virtual squid that we have control over its behavior and showing the squid to a group of Humboldt squid and then making it do certain behaviors and seeing how they respond. Gosh, that would be cool.
I'm also curious of what other adaptations they had to make to be able to survive in such a weird environment.
This habitat is huge and it's mostly dark, and it's also, very low in oxygen. In these deep, low oxygen layers, it's really common for animals to be small and lethargic, but Humboldt squid, they're kind of an exception. They're really active animals, they're really muscly, high metabolic demand, yet they have crazy physiological adaptations that allow them to actively hunt and forage in these low oxygen layers and easily catch all these really lethargic prey items that are down there. And you'll see these rapid vertical transitions throughout a day. And what's happening is the animal is diving down into the deep, low oxygen, cold layers, chowing, coming back up to the surface, catching their breath, and repeating the process.
It's amazing to think that in the deep sea, there are literally millions of Humboldt squid living this fast, aggressive, and as Ben said, often cannibalistic lifestyle, all while continually flashing lights in their muscles and skin to communicate with each other. Who needs science fiction when you got all that going on just a few miles off the coast?
When I say deep sea exploration, you probably get images of intrepid explorers climbing into deep sea submarines and putting on metal diving suits. But much like drones have transformed the way we take to the skies, it's unmanned and autonomous vehicles that are leading the way in ocean exploration.
On one recent expedition to the bottom of the Pacific, the low-kite ROV discovered as many as 30 new species, including this gummy squirrel. Technically a sea cucumber, not a squirrel though.
I'm here at Woods Hole Oceanographic Institution, and this is the Avast Lab. Andy, hello.
Jada, nice to see you.
It is so wonderful to meet you. This place is incredible. There's so much cool technology in here, so what is there that you can show me?
Yeah, well, we're standing in the Avast building, which is dedicated to developing new types of autonomous underwater vehicles as well as associated sensors. So we have a test tank here. We've got a pressure test facility upstairs. We have a rapid prototyping. And so it's about speeding the process of building new tools.
So I see we've got a couple smaller instruments over here. Would you like to tell us a little bit about them?
So this is a very small underwater robot, and this one, has a camera here. It's even got a little mechanical claw for grabbing onto things, and then propellers and lights.
As much as I'd love to spend my time scuba diving on coral reefs, an ROV like this can dive way deeper and stay underwater for much longer than I can. Here's the same type of robot working at over 50 meters with the Hoey Reef Ecology Lab. It's helping them to describe and understand the unique deep or mesophotic reefs of the Coral Sea Marine Park near Australia.
In a sign of how far things have come, now we've got an Xbox controller.
That's fantastic.
I
could use that. That's great. That's awesome.
You could.
How deep can some of these robots go?
This is a very shallow system,
Right.
intended to be used in a tank here.
Right.
And then we have a range of devices, vehicles that have a whole suite, if you will, of depth capabilities. This is our vehicle Orpheus. Unlike the robot we were looking at before, this vehicle can go over 7 miles down in the ocean, actually, 11,000 meters.
A quick reminder of what we're dealing with here. Plants and scuba divers can't survive past a few hundred meters. At 1,000 meters, it's total darkness. No mammals have been found deeper than 3,000 meters. Orpheus can dive almost 4 times deeper than that.
It's designed to go into the deepest part of the ocean, which is actually called the hadal zone. And so this is typically associated with very deep parts of the sort of ocean basins or in trenches, the greatest one of which is the Mariana Trench in the Western Pacific. But these trenches have basically been virtually unexplored.
So what kind of sensors do you have on this to be able to explore? How are they helping us discover different things about the hadal zone?
Think of it as a little bit of a pickup truck. It's got propellers that allow it to fly, if you will, once it gets close to the seafloor, as well as lights. Got batteries on board, and then has a series of cameras that are not only document the environment, but actually also plot the vehicle's movement across the seafloor.
So what's your favorite thing that you've been able to see with these cameras?
So a number of years ago, before we built Orpheus, we had a much larger, more complex vehicle. And I can remember when we went to the Mariana Trench, it's actually deeper than Mount Everest is tall. So it's a little like climbing Mount Everest, getting to the top, but we were able to turn to the scientists and say, gosh, we're here now, what would you like to do?
Right.
There is life there, adapted to such an incredible, harsh environment. And there's science that really suggests that some of the adaptations that life has made to live in the hadal zone actually informs us about life that might exist on other planets, which is kind of mind-blowing. So Orpheus is going to go off next year to the Aleutian Trench, fundamentally just explore. Every time we look in a new part of the ocean, we discover something surprising. Just tells you how much we have to learn about the ocean.
As a scientist, I thank you for all the work that you do in helping to build these amazing things.
Thanks very much for visiting today, and onward, as I like to say.
Onward and I guess downward?
Yeah, there you go.
Obviously, there is a lot to see at Woods Hole Oceanographic Institution's Avast Lab, but they also have some collaborative efforts with Monterey Bay Aquarium Research Institute. So let's head over there to see what they can show us.
Hello, Kakani. So we've been looking at the roles of AUVs in the deep ocean, and I've been doing a little bit of reading on this deep PIV system. Can you tell me a little bit about what it does and what challenges it tries to solve?
My group is called the Bioinspiration Lab, and what we focus on here at MBARI is developing imaging technologies so that we can see life in completely different, novel ways. The imaging instrument that's right next to me is called Deep PIV, and so this is that instrument that can go on remotely operated vehicles and is rated to 4,000 meters deep. It involves an imaging system and an illumination system. And in our case, it's a laser that we're using for illumination.
Photos and high-def video are a great way to study an environment. But these rely on having light, and we're still working usually in 2D. Kakani's team is taking a new approach to this. They're using lasers to scan in 3D.
So the first thing is a control can, and so within this control can you can find a laser, the power supply, etc. And then everything that you see up top is actually what the manipulator arm, right, the robotic arm that's on the remotely operated vehicles actually carries. A high definition camera, video camera system. And then here you can see is the laser housing that has some optics, so we can transition from a point of light to an entire sheet.
And so I understand that you're using Deep PIV to study larvaceans. What is a larvation?
So larvaceans are actually one of my favorite animals. They are what we call basal chordates. So they are very, very, very loosely related to us. And what they do is they make these beautiful, amazing structures that I like to call snot palaces. mucus structures that they secrete from their heads that create these beautiful, intricate shapes that play an important role in filtering food and other particulate from the water around these animals.
That's fantastic. I absolutely love it. How is scanning in the ocean different or potentially better than scanning in a lab?
In our case, the giant larvaceans that we study here, I mean, they're much too big to put in a tank. Their mucus structures can be up to 1 meter or 2 meters across. And so we're unable to do these kinds of observations or scans in the laboratory environment. But I want to say that, DPIV can be used for all sorts of different animals in the ocean, not just larvaceans.
So if you had ambitions to become a deep sea explorer, it might be time to learn some robotics. But as a shark researcher myself, I'm off to think about how this is going to help my work. See you next time. Around 80% of the ocean is unmapped, unexplored, and unobserved. That is a mind-blowingly huge amount of the planet where we just don't know what's there. And there are only so many marine biologists and engineers. But like we're seeing in everything from driverless vehicles to chatbots and video games, artificial intelligence has the potential to do many repetitive and complicated things better or faster than us humans can. And it seems that deep sea research is no different. I'm meeting 2 experts who are going to show us how artificial intelligence is changing research in the deep sea. So I know it's really easy for all of us to get stuck in this doom and gloom mindset about the oceans and about the environment, but I know you've got some pretty high hopes for how AI is going to help us find some new solutions, right?
I do think there are definitely some things like artificial intelligence that will allow us to scale up our capabilities for observing, especially life in the ocean.
So I'm curious, where exactly are we at now with what robots can tell us about the ocean, and where do you hope to get in the future with AI and machine learning and things like that?
So what robots allow us to do, right, is get to places that we as humans can't get to.
There are many challenges that humans face going underwater, but pressure might be the biggest. Our lungs hold about the same amount of air as a soccer ball. At 300 meters, they'd be crushed to the size of a tennis ball. And if you dived to the Mariana Trench, they'd be squished to the size of a pea.
And one of the ways in which, right, the robots can be really helpful is that we can do really quick observations, generating data, either imagery or video, right, noting all the animals that are there. But where I think we're trying to go is to use robots to say something about animals, not only if they're present or absent, but also their behavior. What can we learn and understand about their movement to help perhaps one day predict where these animals might be and how that relates to the environment around them.
I've been reading up a little bit on this ML tracking project, and it's a fantastic example of how we can use machine learning in science. Can you tell me a bit about that and what it does?
One of the challenges that machine learning tracking is trying to solve is, providing this capability for scientists to observe an individual animal for as long as possible. So this is an awesome example I love to share. The blue circle indicates the animal that we're currently tracking. And what you'll notice is that animal we're tracking, which is originally a salp, winds up being bumped into and visited by a squid. And then that blue circle transfers onto that squid. So that's what we're tracking now. And then shortly after this whole thing happened, that individual squid then is joined by a whole bunch of its friends. And somehow, some way, we're able to restore tracking on the original individual. But the problem is, from like a robotics perspective or a mission perspective, right, we don't want to lose tracking on that individual animal. And so what we're trying to do, right, is be able to have smarter algorithms, right, smarter vehicles that can distinguish between different kinds of animals or different kinds of objects that we want to study, and have that information then inform what the vehicle is going to do. And the way that we're able to do this is thanks to a massive database called FathomNet. We're able to aggregate this information and then train algorithms to distinguish between different animals that might be underwater. And so what that looks like is not only can we have these vehicles, right, go off and track something, we can now have vehicles that are smarter, that can go out and search for animals that we actually want to study or learn more about. And so this video I'm about to show you is a field trial where we're doing that. We're demonstrating these ML tracking algorithms. And the top left indicates what mode the vehicle is currently in. So it's currently searching for a snot palace.
A snot palace is a common name for the mucus structures that larvaceans build. A larvation is a free-swimming invertebrate, which makes large mucus nets to capture nutrients that are suspended in the water.
As the vehicle gets closer to the object, If it matches the class or the object that we want to study, you can see we transition to acquire, and once it's been verified, we are now autonomously switched to a tracking mode. So imagine someday we can have these robotic vehicles go out and search for animals that scientists want to study, or even search for animals that we don't know very much about. And that's all being enabled by artificial intelligence on these robotic vehicles.
Now, we focused mostly on the engineering challenges, but I want to find out more about how this is being used in practice. Over in the UK, Carrie Howell is a professor in deep sea ecology and a pioneer in this technology. It's great to meet you, Carrie. Can we start with a quick summary of your research?
So we map life in the oceans, and we do that by basically looking at where animals like to be, the sorts of environmental conditions they like to be under, and then we can use complicated maths to then predict where else those animals are likely to be. And that allows us to produce modeled maps of vast areas of deep ocean for which we just don't have any data. We use things like, well, autonomous vehicles and also remotely operated vehicles as well to go down to the seabed and film or take photographs of the animals that live there.
So how much data are we really talking about here? And what's the difference between a human and a machine learning tool analyzing this data?
A single AUV dive of something like 22 hours will return over 100,000 images. And that's where artificial intelligence comes in. We get the machines to try to identify the animals for us. And that makes that process very, very much faster and means that as a biologist, I can then take the data, which is what I really need, and start putting it in my mathematical models to make my maps.
So if I'm understanding this correctly, it allows you to produce highly accurate maps without necessarily surveying every location?
Well, that's the real trick, and it comes from having enough data to train those models to know that this is a starfish, or this is a crab, or this is a fish, and even more detailed than that, very specific species of starfish. And so we need those large training data sets to make the models work. And some of the work of the FathomNet program is absolutely crucial in that. So collecting all of those annotated images together in one place that come from different laboratories so that we have a big enough set of data to enable us to train models so they work really, really well. The future is looking really exciting for this sort of work, because actually what would be even better is if we could put that AI on board the vehicles themselves, so that when those vehicles are out in the ocean and seeing animals and taking photos and video, that they are interpreting them on the fly. And that means that when those vehicles come back to the surface, they have already got the data. So a scientist can be presented with a data set instead of lots of images that they then have to go back and process.
Just a reminder here, the usual scientific model is to collect data, analyze it, and then present your results. But what we're talking about here is doing all of these things before the robot even gets back on the boat.
Beyond that is actually having vehicles that can respond to what they're seeing. You can imagine if you have an autonomous vehicle, it's gone to a site to check a map. And the vehicle is able to take a photograph, know if it's seeing the animals that it thinks it should be seeing. And if it's not seeing the animals it thinks it should be seeing, it can alter its course to go to another location on the map where that habitat or creature is predicted to be, and check that location instead.
What I really want to know is, can this help with marine conservation?
Well, maps are really critical for making decisions about what you're going to allow to go on where. So if you think about it, if you're being faced with putting a new wind farm in or an oil and gas platform, you know, one of the first questions you want to ask is what's potentially the environmental impact of allowing a development to go ahead? And so at the basis of that is, well, what's there? What habitats are there? What animals, how sensitive are they? So these maps are really important for making decisions about how we spatially use our marine environment for the benefit of society and the wider world. The maps are also really useful for making decisions about where marine protected areas should be.
So if you thought that AI was only for chess computers and smart speakers, think again. AI is also setting sail to explore and help protect our fragile deep sea. If we think of great animal migrations, that probably conjures up images of wildebeest in the Serengeti or humpback whales traveling all the way from Alaska to the tropics. But every single day, our lakes and oceans are home to the largest migrations of anywhere on the planet. It's called diel vertical migration, and it describes the movement of trillions of organisms from the deep up to the shallows and back down again. Diel vertical migration was first noted way back in 1817 when French scientist Georges Cuvier noticed that Daphnia seemed to completely disappear from the surface of the lake he was studying during the day. Years later, US World War II naval warships inadvertently discovered the deep scattering layer of organisms when their sonar reflected weirdly through the midwater. The reflections were very thick and looked to them like sandbanks or even the bottom of the ocean, even though this couldn't possibly be the case. Since its discovery, scientists have been piecing together the data on why and how this happens. Nowadays, we know a lot more about diel vertical migration, but not everything. A world expert on this is Kelly Benoit-Bird. So as a diel vertical migration expert, of course I have to ask you, do we even know why this happens?
Probably the most important reason is that they're trying to balance getting enough food to eat while avoiding becoming dinner themselves. There aren't very many places to hide in the ocean, and so animals stay deep in the dark waters during the daytime to avoid predators that are looking for them. But of course they still need to eat, and food is most abundant in surface waters where light allows plants to grow. So animals move to the surface to forage under the cover of darkness at night before moving down deep in the water column to hide again during the daytime.
So from my understanding, we're not talking about just one species doing this, and we're not talking about this happening in one single area, is that right?
That's right. Vertical migration happens throughout the world's ocean and is carried out by tiny animals to large ones, from zooplankton to squid and fish. But probably the migrator we know the most about are these relatively small fish like this lanternfish, or the bristlemouth fish, which is the most abundant vertebrate on our planet.
Lanternfish get their name from the bioluminescent photophores on their body and heads. They use these lights in the depths to communicate with each other. As Kelly said, these are one of the most common fish in the entire deep sea. Scientists estimate that they make up around 65% of all deep sea fish biomass, weighing in at around 16 gigatons. That's 16 billion tons of glowing fish, weighing more than the entire human population combined. Even crazier is that bristlemouth populations are said to be in the order of quadrillions of individuals.
Each and every day, these animals go through these vertical migrations, making it the largest net animal movement on our planet.
And I'm assuming that it has some pretty serious implications for us and how we understand the ocean, right?
That's true. So the ocean has buffered us from the worst effects of climate change, taking up more than 90% of the heat and about 25% of the carbon dioxide that we generate from burning fossil fuels each year. So plants at the ocean's surface take carbon dioxide out of the atmosphere and use it for photosynthesis. If that carbon can get brought to the deep sea, it gets trapped and can't be re-released to the atmosphere. We're really just starting to understand how vertical migration plays a role in that. It may account for as much as 50% of the transport of carbon from the surface to the deep sea. I like to think of it kind of like a biological conveyor belt. These animals are eating at the surface, migrating down hundreds of meters where they're releasing their waste product, and rapidly moving that carbon out of the atmosphere. It's really important for the health of all of us on the planet.
This is also known as the biological pump. Scientists estimate that if this carbon sequestration didn't happen, the carbon in our atmosphere would be as much as 200 parts per million higher, rendering the Earth completely uninhabitable. Diel vertical migration is fundamental to Earth's natural rhythm, and we need to know as much about it as possible. That's why scientists like Kelly are using all sorts of techniques to better understand how these animals are moving and why. So Kelly, as I understand it, we usually take data and then analyze it later, but you are pioneering a way to take live data of these diel vertical migrations. Can you tell me a bit about that?
We're really fortunate here at MBARI to sit at the head of Monterey Canyon, which is an incredible feature. It's deeper than the Grand Canyon and brings these animals quite close to shore where we can really study them. We've been working with a cabled observatory called Mars. This cable is about 35 kilometers from shore and gives us network and power access so that we can use our sensors continuously. One of the ways we've studied vertical migration is by using sonar or sound, where we send out a short pulse of sound and then we interpret how that sound echoes off animals within the water column. Now, using the Mars Observatory, we're able to take that information continuously so that we can start to understand how vertical migration changes from hour to hour, from day to day, and even over the course of years.
So, from what I'm hearing, this isn't just an up and down, every day, every night type of movement. You've figured out that this is more dynamic and it's changing, right?
Well, we're learning that animals are really finely tuned to the presence and behavior of their predators, which really isn't surprising because survival is so important. But we're learning at what timescales they do that. Vertical migration doesn't happen the same way every night. Sometimes we'll see no migration, and then suddenly a predator will appear, and in the middle of the day, all of the animals will migrate away from the surface and start their vertical migration pattern. Other times we'll see vertical movements at much shorter timescales than that. Juvenile hake, for example, respond to the presence of Risso's dolphins within seconds of us being able to detect them, diving tens or even hundreds of meters deeper, bringing them out of the depth range that these predators can dive to and really protecting themselves. So there's this delicate dance between predators and prey in the pelagic.
It is an amazing phenomenon, but to truly understand what's going on, you have to get in amongst this migration. And not far from here is a robot I know Kelly has used, and it does just that. I'm off to see Mesobot.
So Mesobot obviously doesn't look much like a fish, but it is designed to behave like one, right?
Well, more precisely, it can behave like all of the animals that migrate, that go from the deep ocean during the day so they can stay out of the bright light, but come to the surface to feed at night. So that's fish, it's invertebrates, it's all kinds of different animals. So yes, it can behave exactly like that because it can actually measure those very dim levels of light, and then it can act on those to keep the light level the same. So as the sun is setting, it will rise up just like the fish and the squid and all the other animals do.
Right.
And then at daybreak, it will descend down into the ocean. And so typically it'll go between maybe 50 meters and 500 meters. It's literally holding that light level constant, which is a crude model of what we think the animals do.
Here we see Mesobot's cameras, 2 stereo cameras, and then this is what we call our science camera. Conventional propeller, but these turn very slowly. Radiometer. It can measure the ambient light levels, and it can measure them even when they're extremely dim. This is an acoustic beacon. This is a strobe light, a GPS receiver, and a satellite phone.
So how exactly does your work fit into the whole idea of climate change and the carbon cycle?
Climate models for many years have really neglected the role of the ocean or had very simplified models for that super important contribution. So the more we can understand those processes better, the more we can quantify them, the more we can improve our models and understand how the whole big system actually works.
So Mesobot is helping us to get all of those measurements, learn more about these animals, and understand those processes better.
Right. The more we can understand the daily lives of these animals in detail, how they respond to the light levels, how they move, how long they stay where they are staying, the better we can understand the role of the ocean in climate change.
So there you have it. We have the largest animal migration on the planet and a huge driver of Earth's carbon cycle happening every single day in our lakes and oceans.